So hello, everybody. Welcome to our session where we're going to discuss about how important is using environmental DNA to test biogeographical hypotheses. And I'm here with my colleagues, Mary Edwards, Luke Holman, Jamie Wood, Lea de Nascimento, and Nichola Rius. That is, today he cannot make it here, but he has been as well organizing this session. And when we organized in this session, I had the idea of how important is using environmental DNA. We got a little bit of inspiration from Humboldt himself, because Humboldt is famously quite recognized to having been used a lot of advanced scientific instruments to help advance in biogeography. And what we see in the background of this slide is some examples of his great work on elevation patterns and trying to understand how vegetation particularly has been is distributed in gradients, and in particular these images I put it here. It's Chimborazo in Ecuador, and as well Teide here on the bottom, and his representation of the different elevation gradients. But what really struck our interest was a lot of his writings, because he suggested that, for instance, I should collect plants and fossils and with the best of instruments make astronomical observations, and all the observations that he was able to do on these remote places that he was able to explore. However, he had as well some frustrations. And as you can see here on the top, he was quite frustrated about traveling with these amazing instruments and quite novel at that time, like the barometer that you can actually see here. It looks like Humboldt was quite stressed about all these instruments got lost or broken, as the case of the barometer. And he was like saying that he was quite annoyed sometimes of how painful it was to suffer accidents with this instrument, because of course that means that probably part of his observations were not able to be taken. So following as well with Humboldt and all his traveling, we really like these images when it shows all the way of how he approached these problematics of trying to understand the world at that point in time. And he said something quite nice, that is, lucky are those who travel without instruments that break, without dried plants that get wet, or without animal collection that rots. So in our case, in this session, we will focus in ancient and environmental DNA. And what we think is that the use of next generation sequence is not exception, it's an exploratory for now methodology, but we really think that Humboldt would have been amazed by advances in genomic and the applications with environmental and ancient DNA. For instance, new ways of studying biogeographical patterns, and for instance as well to follow these processes across time, apart from across space. And this is just a pic of the University of Southampton ancient DNA lab, where we have here Charlotte Clark and Tony Brown on the background. So our speakers today, the first one will be Mary Edwards, that he will talk about the challenges facing in terms of environmental DNA for answering the 21st century questions in biogeography. And this talk in particular is work done by Mary and Charlotte, where she will be showing how sedimentary DNA promises a chance to follow processes over time with a really very good detail. More in Mary's talk. This talk will be followed by Luke Holman talk that is about marine biogeographical boundaries across the tree of life. And finally, we will end this talk with this session with Jamie Wood from New Zealand, where he will be introducing his work and the team in New Zealand. And the title is Who Ate All the Truffle-like Fungi? Using environmental DNA from coprolites to test a co-evolutionary hypothesis regarding New Zealand's bizarre truffle-like fungi. So the organization of the event today is this. So Mary will start 15 minutes, followed by Luke, Jamie, and at the end, and this is what we would really like to have participation, is that we think that we are now at a very good point to start discussing questions about how to use this tool to answer biogeographical hypotheses. And we hope to have 15 minutes or even longer to discuss all that in a roundtable. Before starting that, we will share how we think that would be the best way to use the technology to be able to participate. Okay, so just ending with this sentence, lucky are those who travel the world to see with their own eyes, trying to understand it and recollecting the sweet emotion that nature inspires. And I will now stop and I will introduce Mary in this session.
Thank you very much, Sandra. It's great to participate in this session. Can everybody hear me and see me? Sandra, you should monitor in case somebody goes.
All good, Mary. Fantastic. All good.
That's super. Okay, well, I'm going to start out with a very, very brief overview of sort of some of the new methods of DNA, merely to orient anyone not so familiar with them. And then I'm going to talk about a few examples. And Charlotte, my co-author, would have been here, except she's got a new job and she can't. So I'm going to include some of her work. So just going to actually try and get rid of myself. Just a moment. There we go. As far as I'm concerned, I can't see myself, which is good, and I can see the slides better. So, as Sandra has pointed out, Alexander von Humboldt was an extraordinary person with an amazing interest in all things natural. And on many of his journeys, not only did he collect, record, monitor, but managed to come up with these insights, these synthetic insights into how nature actually worked. And here's one of his maps, the one that talks about how there are basically altitudinal zones in the natural vegetation on Chimborazo volcano. And interestingly, Sandra has put up the same photo, and I have cheekily taken the one from the Alexander von Humboldt Foundation, where you might see that he's got a new piece of kit in his hand. And given his tremendous ability to publicize what he did, I think that he would have loved to have been on Twitter. And if he had been alive today, he would have used social media, I think, to decry the state of the natural world, because if he were here today, I think he'd be appalled at the difference between levels of biodiversity today and those that he found on his journeys 2 centuries ago. So modern biodiversity is in a very bad state, as we just heard from the UN today. And much of what we understand about the changes and the mechanisms of biodiversity loss come from concepts of biogeography. And nowadays, many of those concepts are also very important in attempts at conservation and restoration, such as fragmentation theory, corridor theory, and so on. So some of the things that are of interest today to biogeographers and ecologists working with our biodiversity might be, for example, trying to detect the presence of rare species or cryptic species, very, very hard to find by contemporary or normal monitoring methods. Here we have the American bullfrog, which is also invasive in Europe, for example. And then the whole idea of process through time, as Sandra talked about, where we have, for example, refugia, postulated from observations of modern distributions, modern genetic patterns and modeling. Are they actually refugia? What has happened in those places through time? And then following on from that, migration, say, out of refugia or due to environmental change like climate change, we can now, from genetic patterns and combining with fossil data, postulate the places that migrating organisms moved through, but can we actually see what happened to their functional genetics as such changes played out through time? So I think these are some of the questions that in some cases DNA can help answer. So very quickly then, environmental DNA perhaps was first applied in the modern context. And one of the most impressive things was to use it to examine diets of animals that you probably wouldn't just want to walk up and ask what it was they ate. So here, Pierre Taboulet was using analyzing DNA fragments from bear scat to find out what bears ate. So what you have is you have a sample of environmental DNA, or indeed in a minute we'll talk about sedimentary ancient DNA, combined with molecular taxonomies, but not the conventional molecular taxonomies, for example, from sort of the modern taxonomies, molecular taxonomies, which might use very long 500 base pairs, 1,000 base pair fragments, such as the barcode of life, but much shorter fragments that can be found when DNA is degraded in the environment, because as soon as, of course, it gets into the environment, it starts to come apart. Well, so after that, it was realized that you could get DNA out of frozen deposits. So here we have James Hale getting samples from frozen Yedema in Russia. And it's now very successfully taken out of lake sediments as well, for example. So here we have a mixture of, in this case of soil, we have mineral particles, we have biological elements, and the DNA itself is in here often adhering to clay particles or organic particles. So this can be scooped up and analyzed. And by no means try and read everything in this analysis, but just to say that once you've scooped up your DNA, you can basically take it and analyze it in several directions. And a large dichotomy is metabarcoding, whereby you have probably taxonomic or biogeographic questions, and you find fragments of DNA that are hopefully linkable to something like a catalogued flora of your biogeographic region, or perhaps very specific probes that will find a species of particular interest in your series of samples that may be going back through time. So metabarcoding and identifying taxa using a lookup, very specific lookup database, subject to various forms of bias during the process of preparation of the samples, and much has been written about this, and much work is done on it. The other sort of main branch of what one can do is the so-called shotgun sequencing, where everything in a sample is pulled out to a level that is possible, that is, and compared against every conceivable lookup. So the large DNA databases, the large global DNA databases, to see what one can find. And again, one might then apply very particular catalogues to it, or one might try to find as many portions of the genome floating around in that DNA soup as possible. So less bias, but often more uncertain identification, but the potential to cobble together larger genome elements. So these are the ways that one can approach using ancient DNA. So just a few examples of the points that one might follow up using ancient DNA. Rare or cryptic species presence. Within a short time, if you can sample water, you can find the DNA of species that might be very, very hard to find or to see, or certainly to count, or to map in specific habitat areas by searching for their DNA. And so one of many examples of using DNA in aquatic systems to find out whether cryptic species are actually present. And we know that, for example, amphibians are particularly in trouble at the moment all around the world for various reasons, and very important now to try and log their distributions, their presence, and get an idea of how common they are in particular ecosystems. One also might want to focus more on invasive species coming into what might be otherwise unusual ecosystems worth protecting for their their, if not pristine, for their very important biodiversity features. And here we have the Kerguelen Islands in the Southern Ocean, and this study by Pizzitola et al. Very nicely combined, looking for the rabbit DNA itself when rabbits were introduced to this island and started to transform the vegetation on it. Also other elements of grazing such as Sporamiella spores, and then looking at the composition of plant DNA and the erosion rate. And one can see very clearly basically exactly when the rabbits arrived and the major switch in dominant vegetation types, or the species dominant in the vegetation, that was consequent upon the rabbits arising, and also how the erosion rates peaked when the rabbits arise. So one can actually document the consequences and the time pattern of how an invasive species, in this case, has an impact on an ecosystem. And my second example, what actually happens to taxa in what is a postulated refugium over a long period of time. And von Humboldt, of course, went quite a long way into Siberia in one of his journeys. He crossed the Urals near Ekaterinburg and traveled further eastwards into Siberia. He didn't get as far north as this. This is the polar Urals region of Siberia and northwest Russia. In this region, the ice sheet didn't quite get to the polar Ural mountains. There were local glaciation, local glaciers in the mountains, of course, but it was not glaciated in the last glacial maximum. Hence, there are sites in this region which go back a long way in time. And this is Lake Bolsoi Shuchi, which was caught by Jönninge Svensson. And it's a very large finger lake within this polar Urals area. And it's been ice-free since before the last glacial maximum. Jönninge and the team collected a core from this lake that represents that period of time. And the idea that we came up with and Charlotte pursued for her thesis was that such areas as these mountain fastnesses with a lot of elevational variation within the catchment of a lake ought to be able to support multiple plant communities, even as climate changes out of the glacial period into the much warmer Holocene, because of the heterogeneity of habitats and the ability of plants, presumably, to migrate up and down the elevational zones, as von Humboldt pointed out. So the problem with sort of asking what actually happens in a refugium across a long period of time, do the plants actually stay there, is that pollen analysis itself and macrofossil analysis are limited into the taxonomic resolution and the number of taxa they can find. And it turns out that this particular lake in this mountainous region was particularly good for recovery of DNA, clay-like sediments, a large catchment, plenty of inwash, a river. So this lake is collecting information from a very large and diverse and altitudinally variable catchment. And so Charlotte looked at the DNA coming out of this core with particular question: what do the Arctic alpine flora do? Does it persist from the late glacial all the way through, from the last glacial maximum all the way through the late glacial, through the Holocene? And can we detect it today, where, of course, it's growing high up in the tops of the mountains? One of the advantages that DNA has is it avoids this pollen swamping problem, whereby as soon as trees and shrubs come onto a northern landscape, the Arctic alpine entomophilous flora almost vanishes from the pollen record. And this is a highly stylized summary where each block represents the presence of a particular taxon, usually a species or genus, going from 24,000 years ago to almost the present, with a focus on the Arctic alpine plants. And the intensity of the color is the number of times in 8 repeats that particular molecular taxon shows up in the analysis. So each analysis repeated 8 times. And what we can see is the persistence, by and large, of many of the elements of the Arctic alpine flora, right in even into the shrub-dominated period prior to the arrival of conifers, which happens in the Holocene maximum, and out again beyond that into the cooling phase of the last part of the Holocene. And this bar actually says that within the flora of this catchment today, we find all those taxa. So they have in fact made it from the last glacial maximum right through to the present. And we see a very different pattern for the shrubs and trees being focused in the Holocene. So still staying in the north and staying with the post-glacial migration, this paper by Mari Metatallisfrud et al. Looks in detail at genetic variation in Norway spruce, following, as shown here by this contoured color, the migration out of what were probably refugia in Russia or down in Mediterranean Europe, across to the limits of its current European distribution. And very elegantly, this paper tracked two mini satellite regions in for a certain gene, came up with 28 variants, haplotypes, showing the genetic variation as this species spread through Europe after the glaciation. And what the question is that we might ask is, beyond displaying genetic variation as recorded by the mini satellites on one gene, what has happened to the populations of Norway spruce as it's migrated over many thousands of years and many thousands of kilometers to reach its current range? And what we would need to do for that is to pursue the idea of reconstructing the genome and looking, for example, for functional genes, for traits that might be selected for as the species experiences environmental change in space and time. And recent work by Inge Elsos's group has, for example, been able to use shotgun sequencing to put together the genome of an aquatic alga from the last glacial maximum time. And this then really has tremendous promise, because it suggests that we can link functional genomics, the patterns of spread, the nature of environmental change, to understand how it is that species respond to the pressures of environmental change and how they are able to adapt. So acknowledgements to Inge Elsos, Piet Heinzmann, Yuri Lammers, and Jon Inge Svensson from the Ecogen and Chase projects, which Charlotte and I were a part of. And Charlotte's work was also supported by NERC. Well, thank you very much for listening. And I'm now going to hand you over to Luke, who's going to talk about his work in marine systems. So I hopefully have stopped sharing, and over to Luke.
Lovely. Thank you, Mary. Can you see me all right before I hand off? Is everyone all right? Okay. You can hear me okay. Brilliant. Lovely. Right, let me get a presentation up. Okay. I was jealous of Mary's pointer, so I'm going to put one up there. Can you guys see that presentation all right? Is that all right, Sandra?
Nope. Nope, there's no presentation.
No presentation. Oh, no. What a shame. Hold on a minute.
If you open it first in your desktop, and then you press share screen, it should appear there.
Well, there we are.
Excellent. Fantastic.
Maybe now?
Yes.
Sorry about that.
Brilliant.
Wonderful. Right. So, hello everyone. My name is Luke. I'm talking about and thinking about marine biogeographic boundaries across the tree of life, and this is some work I'm doing as a part of my final PhD chapter in Southampton. I wanted to start off thinking about Humboldt, and that's the theme for today. And I was looking for some interesting passages in some of his work to see if he was thinking about some of the similar things to me. And I came up with this small section from Cosmos. And you can see, don't worry about reading the whole thing. I'm going to read you the key bits that I want you to think about. And the first is that he's saying, the universal diffusion of life across the whole surface of the Earth. I'm going to pull up a laser pointer here. And there, I think he's talking about what we might describe as biogeography, things diffusing across. Here he goes on to talk about with respect to the elevation and depth. So he's thinking not only about things changing their distribution, but how that relates to the things that we can measure. In his case, he's thinking about elevation and depth, but I often think about things like temperature or salinity. And then finally, he's talking about microscopic organisms in the ocean. So I couldn't resist putting this one up because of that. And in this case, he's talking about Ehrenberg's work on diatoms and protists. So you can see 250 plus years ago, Humboldt was already thinking about the same sorts of things that I was in this work. So it's really interesting to see that carry through. So when I'm talking and thinking about marine biogeography, these sorts of images come up. Here on the left is a visualization of a recent designation of realms. You can see some of the ocean basins you might recognize being distinct in this designation. And on the right, you can see a smaller version of that, where you're splitting up those larger realms up into ecoregions. And again, you might be able to spot some of the places that you know, and you might have been to these places and be able to recognize that they're different. Something I think about when I look at these designations is actually most of the species that we're using to build these things are well-known metazoans. They're well characterized, they're well described, they're taxonomically pretty well known. And that's sort of a bit confusing to me, because sort of life doesn't stop where we stop studying it. And these researchers here have noticed that as well. This is a brilliant little opinion piece that I encourage you to go and read. It's fantastic work, and these authors argue that microbial ecology and macroecology have sort of been apart for too long, and it's time to bring them back together again and see if we can see if there's any new insight from doing that. And that's kind of what I'm hoping and to show you today, and the sorts of things that I was thinking about when I did this work. And so, do organisms across a huge variety of sizes obey the same rules of biogeography? So Mary really nicely introduced environmental DNA for me, but here's my slide to talk about what I think it is in the marine world and the aquatic systems. And it's everything in this little green dotty box here: tissue, cells, bits of cells, extracellular DNA. It's not one single thing, it's a group of things. And when we isolate this environmental DNA in marine and aquatic systems, we use a filter. So we'd filter water. And here's a scanning electron microscope image I took of one such filter. It looks a bit like a Swiss cheese. You can imagine the water rushing through. And then here's my image after you've taken your sample, and you can see some diatoms in there. Where's my pointer? There it is. And you can see all sorts of bits and bobs of fragments. And I can't myself figure out exactly which parts are environmental DNA. So I'm going to call it all environmental DNA, what you see there. And I also wanted to explain metabarcoding too quickly. I think Mary did quite a good job. I'll just do this quickly as well, so you've got an idea of what the sort of method I'm using here. All these organisms, including bacteria, produce DNA that's sort of present in the water column. We're then able to take that, isolate it as I described, and then sequence it, producing a data set that looks something like this conceptual diagram with a number of sites and lineal columns and a number of species in each row, and some rough measure of proportional abundance of those organisms. In this case, we've only got 5, but many typical metabarcoding data sets would have hundreds or even thousands of different species for their analyses. So the aims for this work, I wanted to see if the biogeographic patterns that I observe apply across the different domains in life. And I was also interested to see if I could explain any of these patterns using what we know about the abiotic or the biotic conditions of the region. And so to do that, I went to South Africa, which it's got this amazing system with this very cold upwelling ecoregion on the west coast, and a very warm system on the east coast, and a sort of intermediate system on the south. And there's been lots of work to think about these regions and how they relate to each other. So it's great for this kind of experiment. It's also an interesting coastline because it's had a lot of shipping pressure, and not just in sort of modern days. This is an image of sort of all the ships that were going around the coastline. This was before COVID. I'm not sure how it looks at the moment, but it's also historically been quite an industrial coastline. If you wanted to take goods from Asia to Europe, you had to go through the Cape of Good Hope, which is down here. You have to travel all the way around. So it's a region that's had a lot of human pressures continuously for a long time. So I went to 18 different locations across this coastline, and here's a a map to give you an idea of the sorts of places I went. Don't worry too much about the differences between the icons, the crosses and the circles. They're designations of whether the site was sort of more artificial, like so a marina, or more natural, but I'm not talking about those analyses today. And then I did some eDNA metabarcoding of 3 replicate water samples per site, and I sequenced them using 3 different genes, and I'm going to call them CO1, 18S and 16S. Now the key thing there is to realize that your CO1 and your 18S data generally corresponds with your larger organisms, your things that you might recognize as sea creatures, and some things you might not, smaller diatoms and single-celled organisms. Whilst your 16S data usually, and in this case, refers to bacterial organisms. So think about that when we're going through some of the charts. And here's some small section of the bioinformatics that we used in this project. I'm not going to talk about that. You don't need it to understand the results, but if anyone wants to talk about those, I'm happy to chat about that later, or maybe in the question section afterwards. So what did we see when we went across this coastline, collected these samples? And here's a non-metric multidimensional scaling based on Jacquard dissimilarity. And the way to interpret this is if your points are close together in these 2 dimensions, it means the communities of those 2 sites are more similar. If they're very far away, they're very dissimilar to each other. Don't worry about the names here, that's just to help me. But what you should look at and see, hopefully, is that the colors of these sites here have matched up with the colors of the ecoregions of the South African coastline. And what I think you can see, and I hope you all agree, is that we're clearly separating these 3 previously described ecoregions out using the eDNA data for the CO1 and also for our 18S. So we can see our larger organisms, they're all sort of obeying the patterns that we'd expect. But when we go to a 16S, we see exactly the same thing. Yeah, also the bacteria, the thing that seems to be structuring their communities along this coastline are those ecoregions. So that's very interesting. And if you take this data and you put it into a sort of modeling structure here at Permanova, asking the question, is there a significant difference between these ecoregions for each of these different markers? And yes, you do get a significant result in that case. So that's very nice, but I kind of wanted to go a bit further and say, well, yeah, there's some patterns, but can we explain any of these observations using what we know about the system? And so to do that, I went and got multi-year average values of temperature, salinity, chlorophyll, and a composite measure of human impact that sort of includes things like pollution and local buildings and invasion potential. So it's a sort of composite. And these are multi-year averages are taken from satellite data and in situ observations. So it's getting over sort of seasonal or year effect. I then performed a distance-based redundancy analysis, which is an analysis where you try to take the dissimilarities between from your eDNA data and try and explain that variation using a number of variables. In this case, the variables are just explained. And so each of these columns here represents a model that I've generated using a number of these variables. And you can see that the models that I use to explain this variation with only a single variable, they explain less of my variations you might expect. And as you go up with increasingly complex models, you explain more and more of variation. So what that shows me is all these variables have something to add to the story, but and individually they explain a different proportion of the variance. But of course, there's some autocorrelation between your sites. So sites that are close together will naturally have a bit more similarity than you'd expect by chance. So I wanted to see if I could cancel out the effect of geography or geographic distance on this coastline. And to do that, I performed a partial mantle test and also a corrected version of a mantle test, which simply takes a distance matrix of your OTU dissimilarities, your sort of differences between your communities in the system. And it looks for a correlation in a distance matrix of each of your explanatory variables after having taken into account the distance between the sites, so the geographic distance. And the geographic distance here is measured. You just went on Google Earth and drew a huge long line across the whole coastline and measured the distance between all those sites. And when you perform that analysis, what you see is that the observations we saw in the last slide to do with salinity and chlorophyll were mostly actually are mostly explained by geography rather than the variables themselves. So only sea surface temperature and this composite measure of human impact remains significant in this analysis. Here you can see I've taken this NDMS diagram on the left, and I've just used the same points, and I've overlaid the variables here, so you can sort of see and what we expect is found in these charts. So the hotter east coast sites would be predicted to have higher temperatures, and the human impact measures, as we'd expect as well. We don't expect one particular coast to have all the impact, so it's more variable of a surface here. So back to that question about sort of across taxa, and I'm not going to expect you all to go through and check all of these diagrams for to see if it matches. I'll tell you, yes, across all the different taxa and all the different markers, you get identical patterns occurring, which I interpret as a sort of convergent cross-phyla biogeography, which I thought was quite a neat result. So the big three take-home points in my work, I think, here was that there were some really consistent and interesting biogeographic patterns across these taxa, that we can explain these patterns predominantly in this coastline. It's structured by or correlated with temperature and some measure of human impact, which we can further break down in later analyses. And I wanted to add this one. I think we're all in this session pretty sold on environmental DNA, but I think it's a brilliant and powerful tool for doing these kinds of ecoregion and biogeographic analyses. So I'd really welcome any feedback on thoughts. This work's not currently published. This is a brilliant portrait of Humboldt, who famously raced death in his latter years to finish his great compendium, the Cosmos, this huge book that contained a vast number of his interesting observations. Now, this work that I've shown you today is much smaller and easier, so I've managed to finish it and submit it before I die. So if you'd like to give any feedback here on the bioarchive, or you can access this from some of the links I'm going to show you in the next slide, that'd be really great. Or if you'd just like to read more of the analyses, I've scratched just the sort of surface of the work that we did in this piece. So of course, thank you to Sandra and the organising committee for asking me to present some of this work. It's been brilliant to be involved. I'm looking forward to the sort of open discussions. Thank you to my colleagues here pictured on the left for coming and adventuring across this coastline of South Africa with me. And thank you to a supervisor and all the group here pictured on the right, in the Ecology and Evolution Lab in Southampton. Here we are pictured in happier times all together outside the sunny National Oceanography Centre. If you'd like to see more about what I'm doing and where I'm working on in the future, you've got these links here that you can check out. So that's me. I think I have to pass on to Jamie to talk about some New Zealand fungi. I'm really excited to hear what he's got to show us.
So thanks for that. Before I begin, I'd just like to acknowledge my collaborators and co-authors on this presentation. Some of the results I'll present today have come about through work undertaken by Alex Boast as part of his PhD research at the University of Auckland. Todd Elliott from the University of New England has looked at a lot of fungal spores on microscope slides, as I'll mention. And our team here on the right is now embarking on a 3-year Marsden Fund project to study the evolution and ecology of New Zealand's truffle-like fungi. So today I'm going to discuss an interesting little biogeographic mystery from New Zealand. It's pretty well known that New Zealand is a fascinating place in terms of its biogeography. It is the largest emergent part of the mostly submerged continent of Zealandia, and it's been physically separated from Australia for at least 50 million years. The current New Zealand biota is a rich mix of ancient vicariant taxa, as well as lineages that have dispersed here at different times in the past. And this unique geological setting, together with the lack of large terrestrial mammals, has resulted in a biota that's characterized by high rates of endemism, gigantism, flightlessness, ancient and basal lineages, and unfortunately extinction. So the particular group of biota I'd like to talk about today are a subset of the macrofungi known as sequestrate or truffle-like fungi. While most fungi rely on the wind to disperse their spores, sequestrate fungi have the spore-bearing tissues enclosed. That is, they sequester their spores inside them. The result of this is that the wind can't get to the spores to disperse them. Instead, most sequestrate fungi seem to rely on animals for their dispersal. The animals that eat the fungi deposit the spores via their dung. The images here show some typical sequestrate fungi that you might expect in most parts of the world. And as you can see, they tend to be dull white, brown or black in colour. They grow underneath the soil and are strongly scented so that mammals with their good sense of smell can find them under the soil, dig them up and eat them. However, back in the early 2000s, mycologist Ross Beaver noticed there was something a little bit different about the sequestrate fungi of New Zealand. While New Zealand does have the stereotypical dull-coloured subterranean sequestrate fungi, Ross noted that an unusually high number of New Zealand species were much more brightly coloured and actually emerged out of the soil to rest upon the forest floor. Recently, Ross Beaver's observations have been borne out by the first quantitative assessment of traits in New Zealand sequestrate fungi. For fungi that are shades of white, brown, black and grey, shown here on the left of this graph, we find that most of these species are actually shared with other land masses. That is, they probably include fungi that have dispersed to New Zealand relatively recently in its geological history. However, orange, red and purple colours are found exclusively in endemic species, as shown here on the right, and so may represent much deeper evolutionary history on the New Zealand landmass. So this is a really interesting biogeographic pattern, but what might be driving this? Well, Ross Beaver came up with a neat little hypothesis to explain it. His idea was that in the absence of large land mammals, New Zealand's sequestrate fungi had instead adapted to exploit birds and maybe reptiles to disperse their spores. So bright colours and emergence from the forest floor are therefore essentially traits resulting from convergent evolution with plant fruits. And these act to make the fungi much more attractive to birds, which tend to find their food mostly using visual cues. While this seems like a highly plausible hypothesis, there's kind of always been one particular fly in the ointment, and that is it's never really been supported by data. And in fact, it turns out that nobody has ever actually observed a bird eating one of these colourful sequestrate fungi. But perhaps this is not so surprising when you consider that around half of New Zealand bird species have become extinct over the past 750 years or so. With such high rate of extinction, maybe the birds that once dispersed these fungi are now gone. One of the key candidates in our minds for having dispersed sequestrate fungi are the moa. There were 9 species of moa that once roamed New Zealand, and these were the largest herbivores, weighing in at around 20 kilograms for some of the smaller species, but right up to 250 kilograms for the largest. All 9 species of moa became extinct within 200 years of humans arriving in New Zealand. However, we know from rare preserved gizzard content samples that moa ate big red fruits, and so perhaps they also ate fungi that resembled them. Somewhat fortuitously, over the past decade or so, we've discovered a number of rock shelters and caves that contain vast numbers of preserved moa droppings or coprolites, as shown here. These dried specimens contain a snapshot of the food consumed by an individual moa over perhaps a few days. Moreover, they contain diverse mixtures of DNA, including the food of the moa, also things like parasites and gut microbes. And so by studying the coprolites, we hope to determine whether or not moa once ate and potentially dispersed these truffle-like fungi. Our first step was to prepare microscope slides from a small subsample of the coprolites. Using these slides, Todd Elliott undertook a survey of fungal spores in about 80 individual moa coprolites, and he identified spores of macrofungi in around 35% of these samples. However, in most cases, it wasn't possible to assign a definitive identity to the spores. Some were quite distinctive and allowed identification to genus, such as the Cortinarius ones here shown with their little spikes. But most of the spores were tiny and lacked any distinctive morphology. Moreover, some genera, such as the Cortinarius, include both sequestrate and non-sequestrate species, meaning that species-level identification is really critical for confirming whether sequestrate species were being eaten. So with the spores failing us, we instead turned to eDNA for answers. As these moa coprolites are hundreds or even thousands of years old, the DNA is in very low concentrations and is quite fragmented. So to minimize the risk of contaminating the samples with modern DNA from the environment, Alex worked on them in labs dedicated to ancient DNA research. These super clean labs have HEPA filtered air supplies, they're positively air pressure, and have nightly UV irradiation to sterilize all the surfaces in the lab. Moreover, we have really strict suiting up procedures and rigorous controls and processes to further remove the potential for contamination with modern DNA. Alex's first steps involved sequencing a short 18S barcoding region from 19 moa coprolites of 4 different moa species. The sequences obtained included a range of OTUs that were able to be assigned to different macrofungi taxa, shown here on the left. 2 of the taxa do actually include sequestrate fungi species. Cortinarius, for example, includes some of the bright purple coloured fungi shown on previous slides. However, it was not possible to resolve the OTUs to species level based on this short barcode region. And so while these data provide tantalizing evidence that moa might once have eaten sequestrate fungi, they don't actually demonstrate this with certainty. However, there are other barcoding genes available, and in particular, ITS is one that's been widely used for fungi. Now, this barcode's a little bit longer than the 18S one we used previously, and so required very well preserved coprolites for it to work. Alex sequenced the ITS region from 2 coprolites of upland moa, collected from caves at opposite ends of the South Island, one at Hodge Creek here in the north and the other at Takahi Valley here down in the south, actually not too far from the Humboldt mountain range. The sequences obtained from the coprolites were blasted against GenBank to narrow down those sequences that were likely to be from sequestrate taxa. Then the identities of these were further resolved with a phylogenetic analysis that included unpublished ITS barcodes from a range of New Zealand fungal species. And so excitingly for us, both these coprolites yielded DNA from a range of macrofungi species. And for the first time, this allowed us to confirm that moa did consume several sequestrate fungi species, highlighted in the red text here. Interestingly, most of these fungi are purple in colour, and purple fruits, along with red fruits, have been shown to be favoured by other forest ratites, such as the cassowary, and maybe this was the case in moa too. So while this answers one question, it raises another. If the large avian herbivores that ate these sequestrate fungi are now extinct, then are any introduced mammalian herbivores now acting as surrogates in this role? This has important ecological and biogeographical implications, as most of these sequestrate fungi are actually mycorrhizal, and they include some of the most important mycorrhizal species found in New Zealand beech forests. To help us understand this, we undertook DNA sequencing on a number of dung samples from red deer and brush-tailed possum. Two of these introduced herbivores in New Zealand, and these dung were collected during autumn in a New Zealand forest. To our surprise, the majority of mycorrhiza taxa detected were native, so the exotic ones are faded out in this plot. And most of the genera include mainly sequestrate species. So the DNA evidence was backed up by microscopic analyses as well, which showed fungal spores to be abundant within these dung. However, the truth is we still don't really know how effective the dispersal by these mammals is. A glasshouse experiment that we undertook using the same dung samples failed to inoculate any seedling roots with these native mycorrhiza. So does this mean that mammals are ineffective dispersers for these native fungi? Over the next couple of years, we'll be exploring some different techniques to try and solve this riddle. So to sum up, we now have the first evidence that moa did consume sequestrate fungi based on eDNA sequenced from coprolites. This evidence supports the hypothesis that New Zealand's sequestrate fungi co-evolved alongside birds and are adapted for avian dispersal. The dominance of birds and lack of mammals on Zealandia therefore helps to explain the biogeography of these brightly coloured emergent fungi, and being more widely represented in the New Zealand fungal flora than those elsewhere. But moa are not the end of the story either. So we have coprolites from a wide range of other animal taxa, and eDNA analysis of these will hopefully shed more light on the interactions between New Zealand animals and sequestrate fungi over the coming years. And so that's where I'll finish. Thank you.
Fantastic. So, yeah, so now it's time to start the question session. Thank you all for these talks. And I hope that people has a lot of questions. So the way we were thinking to organize this is that if you all look at the bottom of your Zoom webinar, you will see the question and answers icon. You can use that, or raise your hand, and then we will need to change your status from attendee to panelist for you to be able to speak. So how we start? Questions?
Leave a long awkward silence until somebody puts a question in the box for us, please.
What I would suggest is that we kind of start, the speakers can kind of start discussing a bit the main things that that you've been discussing. So I see you all now getting into the screen. That's great. And I see Mary, you can actually, yeah, fantastic. We all here now. That's great. So
yeah. Oh, well, just to say, I enjoyed both the other talks immensely. I was curious about the mycorrhizal links of these truffle-like fungi with the beech forests. And is there any research going on, Jamie, about how the beech forests are faring and whether there's any indication that, for example, in new plantations or something, you don't get infection? I mean, is this a sort of tree growth issue right now that people are worried about?
Yeah, it's an interesting question, and it's probably something people haven't considered. And you know, the New Zealand forests have got this kind of progression through the Holocene. So when we came out of the last ice age, we got podocarps, pretty much the first forest types to spread most places. These have been slowly replaced throughout the Holocene by beech forests. And so one of the ideas about how this slow progression happens is that it's actually the fungi that are having to be spread through the landscape before these trees can then kind of get that dominance over the podocarps that are there. And so whether this progression is still going on is an interesting question. And or whether it's been halted by the fact that we had moa go extinct is another really interesting question. And it's something that we're going to be definitely looking at in this 3-year Marsden project we're starting up in this month, actually. So watch this space.
So what I learned as well today with those great talks is that environmental or ancient DNA, what it looks like allows as well, is getting this great taxonomical resolution that we know that sometimes is very well needed to understand better the ecosystems in the past, but as well when Luke was talking about more environmental DNA extractions. So what I was wondering is that this important increase in resolution will allow us to test some biographical patterns like migrations, as well as Mary as well suggested, but as well we could go a little bit more about talking about interactions of different species occurring in the same time, or these networks on the mutualism between species. Do you think that this could be a good a potential challenging but potentially interesting way of using DNA?
Yeah, if I can comment on that, I think, yeah, I think getting to species level is one of the big advantages of DNA over a lot of conventional proxies. I know, Sandra, you're familiar with pollen, and often it's hard to get it past family or genus level. So having that ability to get to species is very cool. And actually, one of the really exciting things about DNA is the ability to get subspecies into actually populations, and looking at how populations of a species might have changed through time.
Exactly. So I think that potentially one of the ways of advancing could be population modeling and interactions. That, yeah. Any other thought in the marine world, Luke?
Well, it's a good point, I suppose, and I think what was really cool about Jamie's work, that if I understood it correctly, is that we're able to say there's definitely an interaction there because it's a coprolite, and as something like analysing the different leftover remains from all sorts of different organisms, you can certainly say interaction. Whilst there's something a little bit different, I think, in saying, well, because we notice these organisms in this area at the same time, you know, there's a jump, I think, from coexisting to interacting. There was recently a nice paper from the marine world where I'll post it up in a minute to our chat so we could look at it afterwards, but they were looking at some interesting interactions, and they were able to resolve these different networks of organisms appearing through time when they were doing a regular water sample at the west coast of America. And they called them interaction hypotheses, right? So we're saying maybe there's an interaction there. We can see this co-occurrence between certain sets of organisms, but we can't say for certain whether there's an interaction there. I think that's a point that we have to make between the sort of observational studies and the ones like what I understood Jamie's work to be, where we're actually able to definitely confirm there's an interaction.
Thanks all. That was a lot of fun. Yeah, and do post interesting things. Yeah,
that was great. Really excellent.
There is a question coming for Jamie. I don't know if we have time to answer.
Far away. I'm going to go. I've got a meeting. I'm sorry, I have to leave, but I shall.
So Jamie, you have a question. We're still open. So yeah. We're still 30 something people. So eventually there is a question. Lea, just please go ahead.
Okay, I don't know if you have access to it, but I can read it for you, Jamie. Okay. Question for Jamie. Are there any other trace fossils or evidence which could be used to test whether MOA help dispersal of sequestrate fungi? I've seen pollen or vegetation trap in teeth to be used as evidence. Would be the beak contain particles?
Yeah, good question. I think most of the beak material we have from MOA is bone, and so we're missing that soft tissue and the keratin that would have been on the outer surface of the beak. So Although there are a couple of naturally mummified moa specimens, I think at this stage coprolites are probably the best evidence we're going to get.
I've got another question, which is, it was fascinating that you were able to go to the ITS region and basically get longer barcodes. Is that right, Jamie, out of the really well preserved coprolites? I was just wondering how long they were, because it always seems to be a kind of limitation of the method, is that you can't get very long sequences from old stuff. Just how long were they? What was there any sort of secrets to the processing or the preservation of these things that sort of...
Some of the coprolites we've got are as young as 650 years. So, you know, probably some of the last moa that were around. And so we've been able to get DNA fragments of at least 400-500 base pairs out of those. We haven't tried to push the limit to see how much we can get.
That's impressive. Yeah.
We have a last, another question, probably the last one, that is for Luke as well. So, it's here, yes. So, whether ecoregions defined by certain species? If so, do you find these species with your 18S metabarcoding assessment?
Okay.
This was from Marta Turon.
Lovely. Well, thanks for the question, Marta. Yes, if you go to these different ecoregions and have a look, you can see very clearly the sorts of organisms there are. And I'm sure that if we were to do an analysis to ask the question of what species define these ecoregions, we would come up with a big list of OTUs or ASVs that would have, in some cases, a species designation, in some cases nothing. That's not an analysis we've done, I suppose. And the second part of your question, whether it's able to be done with the 18S, no, in the case of the 18S, I don't think you'll have enough resolution to get species assessments for many of the organisms across this coastline. I think what I tried to do with this analysis is avoid too much focusing on trying to find the identity of these OTUs. This is a pretty data deficit region in some ways, and we don't have a great number of barcodes or genetic information for organisms that live here. So if you truncate your data by only that you can assign some taxonomy to, you don't end up with very much left to do things with. But I guess that highlights a really key concern as we do these studies. You know, everyone always says, oh, we need better databases, we need better stuff filled in, but you can go to lots of regions today and discover new species without even having seen them. So it's really interesting work to be doing.
Fantastic. That was great.
I just, I'm Kathy Willis, popped up at the end. I'm the current president of the International Biogeography Society, and it gives me a very great, fantastic session. I learned huge amounts, as I'm sure, and I've seen the comments coming through. There are many people saying just fantastic talks and beautifully handled together. And yes, what a celebration of Humboldt and his legacy. I do wonder what he'd make if we suggested that he did away with his instruments and started to use a sequencer instead. But that's for another conversation. But thank you very much to everybody who's joined in, both the panelists, the speakers, and also the audience. It's been fantastic to see you. And please do join us in many other events that are happening this week. Thank you very much. Bye-bye.
Thank you, Kathy. That was great. So thank you all. That was really great. I've enjoyed a lot, and yeah, let's keep chatting DNA, isn't it? Thank you to the attendees as well, that they are here still. So
everyone, thanks Sandra, that was fun.
It was really
fun.
Thanks Jamie for coming from New Zealand as well to join
really
times here.
I'll go up to bed now.
Fantastic. And we miss Marc Rius. Well, I hope that he will see the recording as well.
Good.
Fantastic.
Okay, yeah, send us the link.
Lovely.
Okay, all right.
Bye.
Bye-bye.
Thank you all very much.
Bye, folks.
Thank you.
Thanks, everyone.